Greetings, class, and welcome to our new unit, Chapter 4, A Tour of the Cell. In this particular chapter, we're going to take kind of an overview of the organelles of a typical eukaryotic cell. And then in later units, we'll actually delve deeper, much deeper into some of the organelles. But this particular chapter is going to kind of serve as an introductory unit. And of course, we can't talk about the cell without talking about one of the most important scientific inventions of all time, or technological developments, which is the development of the microscope. Without the microscope, we don't see cells. So it was the development and improvement of the microscope that helped to shape our modern view of the cell and what the cell does and the components that make up the cell. And if you go way, way back to the very beginning of the year, if you remember when we talked about the microscope, we mentioned these two scientists, Robert Hooke and Anton von Leeuwenhoek, who were important historical figures in the development of the microscope and our early understanding of cells and so on and so forth. We remember Robert Hooke, of course, who coined the term cell to describe the room-like structures he saw when he was observing the cork. And then we also talked about Antoine von Leeuwenhoek, who some considered the father of the modern microscope, who developed a microscope that allowed him to see things that nobody had really seen before. And he actually coined a term, we don't really say it much anymore, we call them animalcules, to kind of describe the animal-like microscopic things that he saw. And in this chapter, we'll explore the cellular basis of life. We have 5 big ideas we'll be looking at in this particular chapter. We'll do the introduction to the cell first, and we'll talk about the nucleus and ribosomes. Then we'll talk about the endomembrane system, energy converting organelles, and then finish up with the cytoskeleton. So in our first part of the lesson here, we'll cover part 1 and part 2 here, and then our second video will cover parts 3, 4, and 5. So let's first talk a little bit about the microscope, and I know we talked about it earlier in the semester, but it does bear some review. In class, of course, we use the light microscope, which does allow us to see cells. Remember, light microscopes are limited in how much magnification they can get. They top out probably like 1,000 times. Our microscopes go up to 400, which does allow you to see cells, especially eukaryotic cells, but not making out much of the detail of the organelles that we find inside. For greater magnification and improved resolution, you need to use an electron microscope, either a scanning or transmission electron microscope, which remember we talked about earlier as well. And here's 2 terms that we want to make sure we emphasize again. It's magnification and resolution. Remember, magnification describes how much larger you're increasing the image's size, whereas resolution is describing how clear the image is, how sharp it looks. So magnification goes kind of hand in hand with resolution. If you remember when we did our lab about a month ago with the microscopes, we were able to magnify some of the things to 400, but for some of the images, we didn't really get good resolution. Okay, so the images were larger, but they were blurry. So ideally, you want to capture both. You want to get more increased magnification and increased resolution at the same time. So here's 3 images of a paramecium. This is how it will look when we look at it underneath our microscopes. In the second semester, we look at protists. But if we had a scanning electron microscope, look at the surface area. Now that we're scanning the surface, we can see all those little tiny cilia and stuff. Look at the detail that you can get that you just can't get with the light microscope. And then look at the transmission electron microscope. As those electrons penetrate deep in, you can really make out a lot of the different organelles that we see inside here that we just cannot make out again with the light microscope. So we've got increased magnification and increased resolution at the same time. Now, because of the development and then ultimately the improvements of the microscope, we get the beginnings of what's called the cell theory. The cell theory actually has 3 parts to it. It states that all living things are composed of cells. All cells come from cells. And then thirdly, cells are the basic units of structure and function in living things. So these 3 elements together, I mean, when you think about it, it's kind of blatantly obvious nowadays, right? But somebody had to make these early observations and early discoveries, which were not possible until approximately the mid-1800s, where the microscopes were good enough that scientists could actually start to see cells being born of other cells. Looking at the tissues of different types of organisms and recognize that those were made of cells, like the work that Dr. Schwann and Dr. Schleiden did. and then recognizing that cells actually carry out the day-to-day daily functions of what needs to be done for an organism to survive. And this particular slide is kind of interesting because it allows you to see how you would observe specimens in terms of with your naked eye there, unaided, what kind of things you can observe with the light microscope, and then what things would require an electron microscope to really make out the details. And you can see on the back end here, once we hit the electron microscope, we're starting to look at small bacteria, viruses. These are kind of things we cannot really observe with our light microscopes. We would need something far superior to that. And also to be able to see the organelles, we would need an electron microscope. Now in section 4.2, as we start to talk about the cell, one of the first things we need to recognize is why cells are so small. Did you ever wonder that? Why aren't they bigger? Well, there's some practical reasons why. First of all, in terms of the cell, when you think about it, the cell has to move material in and out, right? It has to be managed, that kind of stuff, and it has to be able to grow and develop like all living things. But what's interesting is the faster it grows, the smaller the ratio between surface area to volume becomes. So what that means basically is the faster it grows, the volume of the cell can start to outstrip the plasma membrane or the cell membrane, which is kind of like if you think of a balloon. The balloon, as you're inflating it, you're increasing the volume, but the problem is there's only so much balloon to go around. So if you keep inflating and inflating and inflating, the balloon will pop, which is kind of similar to what could happen with the cell. There's only so much cell membrane to go around. So the faster that the volume increases, it's very difficult for the plasma membrane to keep up. So the cell could actually rupture. But also some other practical reasons in terms of the cell itself. The larger it gets, the more difficult it would become for the nucleus to manage it. The more difficult it would get to be to move materials in and out of the cell. Even though it is a phospholipid bilayer with proteins that do help facilitate the movement in and out, it would become more difficult to move stuff from like the deepest parts of the center of the cell all the way out to the membrane and beyond. It would be difficult to bring stuff in through the membrane and get it to those furthest deep center parts of the cell. The bigger it gets, the harder it is to move material. So how do cells solve that problem? Okay, what they do is they divide, right? When the time comes that it's time for the cell to divide, and we'll learn about that later in the cell cycle, the cell will divide into two, which brings it back down to a more functional size and also increases that surface to volume ratio. Look at this image here. We have a cell here, like imagine this cube is a cell, and then we have a bunch of cells here, okay? 27 of them, in fact, right? But notice they each occupy the same volume. But we have some differences here. In this one here, notice the surface area is only 54 units, whereas in this one, the surface area is 162 units. Because we have 27 individual little tiny cubes, which are like our cells, each one has its own three-dimensional surface area, so it can contact the surface in all different directions. Okay, so we actually have a surface to volume ratio of 6 here, but only 2 here. This particular cell cannot inflate much more, or grow much more. I'm saying inflate like a balloon. It cannot grow much more because it will outstrip the surface of the cell, and the cell could rupture. But in this case, by dividing, these cells have become smaller again. It's more efficient to move material, easier for the nucleus to manage, and we increase the overall surface area, which is important. Certain types of tissues are much more effective when there's more surface area contacting the surrounding environment. Like, for example, tissues that are responsible for exchange of oxygen, like in a root system or something, capturing moisture from the soil. The more surface area that can contact the moisture, the more moisture can come in. And in the case of like your lung tissue, the more lung surface that can contact the air, the more efficient the exchange of gases will be, or in the gills of a fish, say, or something. So this cell addresses this issue of maintaining a good volume, the surface ratio, by dividing. Here's a cross section of a cell membrane, and we describe this cell membrane. We call this the fluid mosaic model, which we'll talk about in the next chapter when we get more detail about the cell. But basically, you see the plasma membrane is then kind of embedded with all these like structures and proteins and things like that, and channels that will facilitate movement in or out of the cell. How all this stuff works will be the content of our next chapter, chapter 5. Now, before we actually get into the content, we have to differentiate the two different types of cells. We have those cells that are prokaryotic and then those cells that are eukaryotic. Prokaryotic cells are structurally much simpler than eukaryotic cells. They do share some things in common, both types of cells. They have a plasma membrane, DNA, ribosomes, and cytosol, or cytoplasm. The two lines of prokaryotic cells are bacteria and archaea, and then the other line is eukarya, which contains things like plants, fungi, protists, animals. What makes them different, though, is a couple of different things. First of all, eukaryotic cells are distinguished by the fact that they have a membrane-enclosed nucleus and also membrane-enclosed organelles, which you don't find traditionally in bacteria. And additionally, prokaryotic cells are much smaller and simpler by the fact that they don't have all those organelles. Bacteria are much more difficult to observe under a light microscope. That's why later in the year when we study prokaryotes and bacteria, we'll use our microviewers for that. There's an example of a typical bacterium here, and you can see, look how few organelles. I mean, there's not many structures that we find in the bacterium. We see the chromosome here, but notice it's not inside a nuclear membrane at all. And we don't see many other structures. Some ribosomes to help facilitate assembly of proteins, but just not much else, which is a lot different than a typical eukaryotic cell. In this particular chapter, we're going to concentrate on eukaryotic cells. We'll get to prokaryotes later in the year. Eukaryotic cells are partitioned into little tiny compartments. Those partitions are based on these organelles. And of course, the organelles, that means little organs, are the structures responsible for helping the cell do the things that it needs to do to survive. Because remember, cells are living things. Organelles can be divided into 4 basic functional groups, and this is how the chapter is going to lay out. First, we'll look at the nucleus and ribosomes. Then we're going to look at the organisms that make up the endomembrane. Then we'll look at the organelles that are responsible for processing energy, and then we'll finish up looking at the structural support components of a typical cell. Here is a typical eukaryotic animal cell, and look at all the structures that we see here. We just don't see this many structures in a prokaryotic cell. Same goes with a plant cell. Another example of a eukaryotic cell. Look at all these structures here. So let's start first by looking at the nucleus and the ribosomes. And in this part of the chapter, we're going to be a quick overview or tour of the major organelles of a cell. Remember, the cell has 3 distinct regions. We have the membrane on the outside. On the inside, we have the nucleus. And then surrounding the nucleus is the cytoplasm. So we talk a little tiny bit about the membrane, not too much because we're going to talk about that in the next chapter. And now we'll talk a little tiny bit about the nucleus, although again we'll be doing that in a separate unit as well. And then we'll concentrate on the organelles found inside the cytoplasm in part 2 of our video. The nucleus, of course, contains the cell's genetic information. The nucleus houses the cell's genetic information in what's called DNA, deoxyribonucleic acid. The nucleus is also like the control center of the cell. It contains a structure called the nucleolus, which is found inside the nucleus. And the nucleolus is responsible for helping us create the components of the ribosomes, as well as directing the cell cycle, which we'll look at in a couple of chapters. The nucleus, and I'll slide up to this next page here. The nucleus looks kind of like this. You can see it here in an electron microscope image. You see all this spaghetti stuff here. That's the chromatin. The chromatin is the DNA plus a protein called histone. So it's kind of like a messy, squiggleless spaghetti here. And it takes on its more traditional shape, like when you think of a chromosome, and you think of DNA that way, when it's getting ready to divide. We can also see the nucleolus here, where the components of the ribosomes are made, and where also the direction of the cell will take place for the cell cycle. And then outside we have the endoplasmic reticulum, which we'll look at in part 2, which contains the ribosomes, which is where protein synthesis takes place. Now, the ribosomes are important because the ribosomes are where protein synthesis actually occurs. So the ribosomes are made of a material called ribosomal RNA. And as I said a second ago, that's where protein synthesis takes place. The ribosomes are located in the rough endoplasmic reticulum, which is located in the vicinity of the nucleus. So if you look at this image here, you can see here's the nucleus, and then you can see the endoplasmic reticulum right here. Notice that this is called rough ER because it's lined with these ribosomes. and that's where protein synthesis will take place. You can see the mRNA, which carries the message from the nucleus. The DNA is where the information is stored, but the DNA cannot pass through the nuclear membrane, nor should it. It's very important. That information is carried out to the ribosomes via the messenger RNA, and then that information is translated into a building protein. And then that protein is then distributed throughout the endoplasmic reticulum and then delivered out to another part of the cell, which we'll be learning about in part 2. Okay. And actually, that's a perfect place to stop for today. And in part 2, we'll learn more about the endoplasmic reticulum. We'll also talk about the Golgi complex and their relationship together, and lysosomes and structures like vesicles and things like that.